Welcome to this video on Frederick Griffith experiment, discovery of transforming principle. Frederick Griffith conducted a pioneer experiment in 1928 that provided the first evidence of bacterial transformation. He demonstrated that a non-pathogenic R strain of Streptococcus pneumoniae could be transformed into a pathogenic S strain when co-injected into mice along with heat-killed S cells. Griffith concluded that a transforming principle from the dead S strain was responsible for this change. Although he could not identify the chemical nature of this principle, his work laid the foundation for future discoveries and ultimately revealed DNA as the genetic material. Griffith conducted a series of 4 experiments. In experiment 1, he injected non-pathogenic bacteria or R strain into mouse, and he found that the mouse was alive and he was able to isolate the R strain from live animal. In experiment number 2, he injected the pathogenic S strain into mouse and he found that the mouse was dead and he was able to isolate S strain from the dead animals, suggesting that S strain is pathogenic. In experiment number 3, he heat killed the S strain before injecting into mouse, and the result was mouse was alive and he was not able to isolate any S strain from live animal in this experiment. In experiment number 4, he mixed R strain with heat killed S strain before injecting into mouse, and he found that the mouse was dead and he was able to isolate both R and S strain. So this was the first experiment suggesting transformation of R strain into S strain in biological science. He concluded that there was something from the dead S strain which transformed the live R strain into a virulent S form. Although the chemical nature was not identified by him, he called this factor as transforming principle. These findings were pretty significant because he was the first to demonstrate transformation, and it has implications in biotechnology because transformation is normally used in genetic engineering. However, it was also a challenge for the protein-centric inheritance because at that time this theory was pretty dominant. So he challenged in a way, but he could not identify the chemical nature of his transforming principle. However, it laid foundation for identifying genetic material. So thank you very much for watching. I hope you understand the importance of Griffith experiment. Please like it, share it and subscribe it. Until next time, you take care. Bye bye.